Good morning, students. I am Professor Poonam Gharge, going to present a lecture here on historical development of plant breeding. Several significant contributions have been made in plant breeding by various workers from time to time. In broad sense, history of plant breeding can be divided into four parts. First, pre-Mendelian era, before 1900. Second, Mendelian era, it starts from 1900 to 1920. Third one, post-Mendelian era, it started from 1921 to 1950. And the last one is modern era, that will start after 1950. So one by one we are going to see the history of plant breeding in these four parts. Before start of plant breeding, the practice of plant domestication is started and it is estimated to date back 9,000 to 11,000 years. So many crops in present day cultivation are the result of domestication in ancient times. About 5,000 years ago in the old world and 3,000 years ago in the new world. In the Neolithic period, domestication took a minimum of 1,000 years and a maximum of 7,000 years. Today, all of our principal food crops come from domesticated varieties. Cereal crops were first domesticated around 9000 BC in the Fertile Crescent in the Middle East. The first domesticated crops were generally annuals with large seed or fruits. These included pulses such as peas and grains such as wheat. So, domestication is the process of bringing a wild species under human management. So, domestication is considered as most basic method of plant breeding. Domestication is continuously going on today and is likely to be continued in some time in future. So during the long period of historic cultivation, natural selection has definitely acted on the domesticated species, and the movement of these cultivated species started with movement of man. When this man or human beings are going to move from one place to another, they brought the movement of this cultivated plant species. First, pre-Mendelian era. In 9000 BC years ago, first evidence of plant domestication was found in the hills above the Tigris river. Then, in 3000 BC years ago, domestication of all important food crops in the old world is completed. Then, in 1000 BC years ago, domestication of all important food crops in the new world was completed. Then, 700 BC years ago, Babylonians and Assyrians hand pollinated date palm artificially. In 1665, another scientist, Robert Hooke from England, first described the cell, and therefore he is known as father of cell biology. In 1694, Camarius of Germany first demonstrated sex in plants and suggested crossing is a method to obtain a new plant types. Next, in 1717, another scientist, Thomas Fairchild, developed first inter-Pacific hybrid between Sweet William and carnation species of dianthus, which is popularly known as a Fairchild moon. In 1727, the first plant breeding company was established in France by the Villemorians and introduced the pedigree method of breeding. In 1753, Linnaeus published Pesce's Plantarium. Binomial nomenclature of plant taxonomy officially begins with his general list of plant species. In 1766, another scientist, Joseph Kohlreuter of Germany, demonstrated that hybrid offspring received traits from both parents and were intermediate in most traits observed and produced first scientific hybrid using tobacco. In 1800 to 1835, Knight T.A., which is an English scientist, first used artificial hybridization in fruit crops. In 1819, Prof. utilized pure line selection to develop a new oat cultivar as well as a new wheat cultivar. In 1823, Knight again observed dominance, recessiveness and segregation in peas. In 1840, John Le Coiter, they observed the concept of progeny test and individual plant selection in cereals. In 1840 to 50, de Villemorian, who is a French biologist, developed progeny test in wheat, oat and sugar beet breeding. In 1856, de Villemorian further elaborated the concept of progeny test and used the same in sugar beet crop. In 1859 to 89, Darwin published Origin of Species and noted that inbreeding, sterility and differences in reciprocal crosses. In 1866, Mendel G.J. from Austria published his discoveries in experiments in plant hybridization, culminating in the formulation of laws of inheritance in Gharge P and discovery of unit factors. Nowadays we call it genes. In 1873, the work of Patrick Schroff was first published and he concluded that only the variation heritable in nature responded to the selection and that their variation arose through natural sports such as mutation, and by natural hybridization means recombination events that takes place during meiosis that will be helpful for a selection process. In 1890, Rimpu from Sweden, he made a first intergeneric hybrid by crossing wheat, that is a triticum aestivum, with sickle cereal, which gives to the birth to triticale crop. Then second part of history, that is a Mendelian era. In 1900, De Vries from Holland, Correns from Germany and Stechermark from Austria rediscovered Mendel's law of inheritance independently. In 1900, Nielsen H., a Swedish scientist, elaborated individual plant selection method in Sweden. In 1903, Johnson W.L. proposed the pure line theory that provided the genetic basis for individual plant selection and also coined the term genotype, phenotype and pure line. In 1908 to 1909, Hardy of England and Weinberg of Germany developed the law of equilibrium of population. In 1908, Davenport C.B. first proposed dominance hypothesis of heterosis. 1908, Shull G.H. from US and East E.M. from US proposed O'Hare dominance hypothesis independently working with males. In 1909, Shull conducted extensive research to develop inbreds to produce hybrids use of F1 hybrids between inbreds in corn breeding. In 1909, Nielsen-Ille given the explanation of multiple factor hypothesis for grain color of wheat by using pedigree breeding and bulk breeding. In 1910, Bruce, A.B., Kibble F and Pillow C elaborated the dominance hypothesis of heterosis proposed by Davenport. In 1914, Shull G.H. first used the term heterosis for hybrid vigor. In 1917, Jones developed first commercial hybrid maize and proposed dominance of linked gene hypothesis. In 1919, Hayes, H.K., Garber, R.J. both gave initial idea about recurrent selection. Also, they suggested use of synthetic varieties for commercial cultivation in maize. Third important part of history is post-Mendelian era. In 1926, East E.M. and Mangelsdorf A.J. first discovered gametophytic system of self-incompatibility in Nicotiana sandry crop. In 1926, Pioneer Hybrid Corn Company established a first seed company. In 1926, Vavilov and I identified 8 main centers and 3 subcenters of crop diversity. He also developed concept of parallel series of variation or law of homologous series of variation. In 1927, Muller produced mutation in fruit fly using X-rays. In 1927, Karpenchenko developed first intergeneric hybrid between radish and cabbage in Russia. In 1928, Stadler L.J. from US first utilized X-rays for induction of mutation in barley crop. In 1933, Rhodes discovered the cytoplasmic male sterility in maize crop. In 1934, Dustin discovered the colchicine. In 1935, Vavilov published the scientific basis of plant breeding. In 1935, Nagaharu Yu proposed the origin of tetraploid species of Brassica using a triangle which is popularly known as a Yu's triangle. In 1936, East E.M. supported O'Hare dominance hypothesis of heterosis which is proposed by East and Shull in 1908. 1939, Golden C.H. first suggested the use of single seed descent method for advancing segregating generations of self-pollinated crops. In 1940, Jenkins M.T. described the procedure of recurrent selection. In 1944, Avery, MacLeod and McCarthy described the transforming principle and suggested that DNA, not a protein, is the hereditary material. In 1945, Hull F.H. coined the term recurrent selection and O'Hare dominance working with maize. In 1950, Hughes and Babcock first discovered sporophytic system of self-incompatibility in Crepes foetida. At the same time, Grestel discovered the sporophytic system independently in Parthenium argentatum. In 1950, McClintock discovered the ACDS system in transposable elements. Then fourth one is modern era. In 1952, Jensen and F first suggested the use of multilines in oat. In 1953, Borlaug N.E. first outlined the method of developing multilines in wheat. 1953, Watson, Crick and Wilkins proposed a model for double helical structure of DNA. In 1956, Flohr, H.H. proposed gene for gene hypothesis for host parasite resistance. In 1964, Borlaug N.E. developed high-yielding semi-dwarf varieties of wheat which resulted in green revolution. In 1965, Graphius J.E. first applied single seed descent method in oat crop. In 1968, Donald developed the concept of crop idiotype in wheat. In 1970, Patel C.T. developed world's first cotton hybrid for commercial cultivation in India, and hence Patel C.T. is called as a father of hybrid cotton. In 1970, Borlaug received Nobel Prize for the Green Revolution. In 1970, Berg, Cohen and Boyer introduced the recombinant DNA technology. In 1976, Yon Long Ping et al. Developed world's first rice hybrid by utilizing cytoplasmic male sterility for commercial cultivation in China. In 1983, Fraley et al. Developed first transgenic tobacco plant in USA. In 1987, Monsanto developed world's transgenic cotton plant in USA. In 1991, Icrisat developed world's first pigeon pea hybrid, that is ICPH8, by utilizing genetic male sterility for commercial cultivation in India. In 1994, flower sour tomato developed as first genetically modified food produced for the market by Calgene Company of California. In 1995, BT corn developed. In 1996, glyphosate resistant crop soybean introduced. In 1997, Monsanto from USA first identified terminator gene which allows germination of seed for one generation only. In 1908, Monsanto identified traitor gene which responds to specific brands of fertilizers and insecticides. 2001, Protection of Plant Varieties and Farmers' Right Act get established. In 2002, Mexico and Monsanto together released 3 BT hybrids, that is MECH12, MECH16 and MECH184. In 2004, glyphosate resistant crop wheat was developed. In 2005, Government of India approved cultivation of some BT cotton hybrids in Punjab, Haryana and Rajasthan. Next, institutional development and history of plant breeding in India. In 1871, the Government of India created the Department of Agriculture. In 1892, first scientist appointed in the Department of Agriculture. He was a agricultural chemist. In 1901 to 1905, agricultural colleges were established at Kanpur, Pune, Saibur, Lailapur and Coimbatore. In 1905, the Empirical Agricultural Research Institute was established in Pusa, Bihar. In 1934, the building of institute damaged due to earthquake. Hence, this building or this institute was shifted to New Delhi in 1936. In 1946, the name of Imperial Agricultural Research Institute was changed to Indian Agricultural Research Institute. In 1929, Empirical Council of Agriculture Research was established in New Delhi. In 1946, the name of this institute is again changed to Indian Council of Agriculture Research. In 1921, Indian Central Cotton Committee was established. Notable researches on breeding and cultivation of cotton was carried out under this committee, and near about 70 improved varieties of cotton were released in this period. In 1956, project for intensification of regional research on cotton, oilseed and millets was initiated to intensify research on this crop. So these centers or these projects are located at 17 different zones or centers throughout the country. In 1957, All India Coordinated Maize Improvement Project was started with objective of exploiting heterosis. In 1960, first agricultural university established at Pantnagar, Nainital, UP. In 1961, the first hybrid maize variety released by the project, that is All India Coordinated Maize Improvement Project. ICAR initiated coordinated project for improvement of the other crops. In 1961, first maize hybrid was released, that is Ganga 1, Ganga 101, Deccan and Ranjit for commercial cultivation. In 1964, first sorghum hybrid, that is CSH1 was released. In 1965, first Bajra hybrid HB1 was released. In 1971, one of the Pioneer Center for Research in India, TNAU, was established. In 1991, first Pigeon Pea hybrid ICPH8 was released from ICRISAT. ICRISAT currently working on 6 crops, that is pearl millet, sorghum, pigeon pea, chickpea, finger millet and groundnut, and it is located at Hyderabad. Now we are going to see the important contribution of some Indian plant breeder. The first one is T.S. Venkatraman. He was an eminent sugarcane breeder. He transferred thick stem and high sugar content from tropical noble cane to North Indian cane. So this process is known as a nobilization of sugarcane. He developed several varieties of sugarcane having high yield and high sugar content. He was director of Sugarcane Breeding Institute, Coimbatore. Second important scientist is B.P. Pal. He was an eminent breeder known for his contribution in the breeding of superior disease-resistant NP varieties of wheat. He was the first Director General of ICAR. Third one is M.S. Swaminathan. He was a famous plant breeder responsible for Green Revolution in India. He developed high-yielding varieties of wheat and rice. He was Director General of ICAR and later Director General of IRRI Philippines from 1982 to 1988. Another scientist is Pushkar Nag. He is a famous potato breeder who developed several high-yielding varieties of potato. He was a director of CPRI Shimla, that is Central Potato Research Institute. Next important scientist is N.G.P. Rao. He is an eminent sorghum breeder. He developed several high-yielding hybrid of grain sorghum. He was chairman of ASRB and vice chancellor of Marathwada Agriculture University, Parbhani. Next one is K. Ramaiah. He was a renowned rice breeder. He developed several high-yielding varieties of rice when he was a director of Central Rice Research Institute, Cuttack. Next one is K. Athwal. Famous pearl millet breeder. He worked in Punjab Agriculture University, Ludhiana and developed several superior varieties of pearl millet. Next one is Bosi Sen. He was an eminent maize breeder. He developed several varieties of maize for hill region of Uttaranchal. Then he established the Vivekananda Parvatiya Krushi Anusandhan Shala at Almora, Uttarakhand and was a director of the same till his death. Next one is Dhampal Singh. He is an eminent oilseed breeder. He released several varieties of oilseed, that is rapeseed and mustard from Kanpur. He was a vice chancellor of G.B. Pant University of Agriculture and Technology, Pantnagar. Next is a C.T. Patel. He was a famous cotton breeder who developed world's first cotton hybrid in 1970 for commercial cultivation in India and therefore he is known as a father of hybrid cotton. Next one is V. Santnam. He is a famous cotton breeder who has developed several high-yielding varieties of upland cotton and 2 varieties of Egyptian cotton. He was the first project coordinator for All India Coordinated Cotton Improvement Project and later worked in FAO as a cotton consultant. So this is all about history of plant breeding. I hope you have enjoyed the listening. Actually, we are not going into the depth of each scientist and his work because from exam point of view, we not require too much details on the research work. So only we are reflected here on the important work of that scientist. So thank you for watching this video.